this Taritalia and take the Katu those Tikatu the Gache. Tati Yapina. Tango catching is enough. The get the take Katu Yayu to disconnect Solea. What? Doxene. We're going to connect, we are going to connect these lines. Those connect che. So biology now remove ya Kevin Yota. Mix a grand so biology link in the Yubina, biology latona Yubina, can cell chick remove ya cell Yubina, they can get Gensha Jire. Tadela remote Tange de Talia, take 10 to Zopa, connect Chevy, link Chevy, those join Chevy. I've connected these lines. Tadus leswa. Tatenenge Kachikisana, Dinane connection and Tadide Chenati. Dinia Chijidu, Suchero. Looks she. Get this social. So make use of your skill. Remote kabu yuna biology na remote kama remote ne jaya shibu yore. So yiki champ kapsi inji meji tikwa tan logo de chigu mare kabre ya remove yune mangbuchi changtwe yore. So remote ya cell chigre de pu changma la yoma de ne yo shab dige de susu cell de ha ko butone dele ya you polish it further. Lob say la ya trowa ke chig tabla mangbu yungre ba. And biology la ya chig ing shue ji ya china zo remote ya bu yobe na di cell the page down beina. You dog chechne sur sama dog cheche I extended this thing. Right? And yet the nangas the pass so chine. And it's not it. Tamatella near the chigi Delia, let me draw line chick line ni. Delia line chick line ni. Line chick line ni dog chechne. Dogs che dogs chene technique gabi ani erchen digdo da tigdi angam taking it de join shi so tigdi tamadini ya tension ya wam sere dogs cherwa de join chi ma che tamadini ya tension de chimogoche outer thick dine dokche and similarly over here denge chimogoche outer dine dokche de la ya doks sarache dokche dokche shirwa ta dele ya doksinda dokse thing yiba digini ya dos chenche and then ma dokse ne dos treche dokse shit. shit. Chita baluchi jangil soi benaya kiranzo tajana la ya tanda dinde yung gure learning style check annotated drawing chechine drawing namgyu annotated drawings na drawing some mind ba drawing la ya deshe yo ba dinde chil sigore kiranzo na taja gi tungen yo be na kiranzo disibe na annotated drawing di kandechire sha sechine kiranzo ya de chilo tarwa ni dine kiranzo pejo tan namgyu ne lobjon chebe kapso so tande tuzo de la ya kechu chik kiranzo la ya moyo kiran dun la ya gegen me du zambal ya rawan chimbishta yore lobju sol ya di tuzo de pe ra khak pore wa tinte indu susu gi di pejo kang mama tang thoye gi tabshi chigurda kiranzo gi independent learning che thoye gi tabshi chigurda dokse ani yete shading chi shorwa remote ge ning je bu soye chido let me shade this thing dokse kiranzo shuga tebe na de ning je sta yung da de ya ma ga pen je da sho chine de zam gi ya wu yung mu do dokse 도시체 drawing 다되네 땅에 같이 있으나 델리아 바로 도할때 숲이로와 디 엔데 치네 갭도 테스트 샤기 다디 고야디야 개좀브레 델리아 갭 딜리아 먼저 줄라 레벨링 체크업 so we call it as plasmodesma seriously so plasmodesma inda so doks chene and chin jon cheje doks che share now these are the plasmodesma right cell nang ya ta ngajo nangi goso ro din ma thye gor ba chala de nangi chala chilo thye gore chilo chala nang la juye gore so this is the channel dil gulam do chre dil lam do chre so doxis theje dosse ta ngajo chilo de thisainda ta nanglo de thiainda wa ta nanglo de ngete chunna manda spestam pena now since this is a plant cell ngajo kechu plant cell na kargolana there is a large central vacuole so I'm going to draw that thing large central vacuole let's say this is the large central vacuole right central vacuole chiche tatene nucleus chichurwa nucleus is surla push share because of that vacuole which lie almost at the center of the cell so nucleus nge di was tv na that's 통계연어와 되는 거지 아니 이게 독시체 안되네 고고로 세우라 these are the nuclear pore and nucleus dia the sik subtatech so like this ki de le se gap do le di ro tong cha di the pore because are so nucleus share nucleus nang lo le ya ni ngan ju gi desher di ungurwa we call it as a nucleoplasm this center thing is called the nucleolus that the nucleolus because da that thing is called nucleolus dokshi che and covering the nuclear membrane the cherwa. We've got nuclear pore, igan the che and nucleoplasm. Thing also jo label chigi. Those che ta nucleus the chabache. Now ta plant cell nang la shen kariyore. Wangbu chungu dende sheba kariyore. Cell organelle sigirwa. Ta cell organelle dige de chik nge di tv na. Those stage those stage ne endoplasmic reticulum tv nucleus ke surlaya endoplasmic reticulum. So endoplasmic reticulum gan la ya nge ribosome khashish thurwa ri diket khashish tiki. These are the ribosomes, machinery that is responsible for making a protein. So let's say these are the ribosomes, right? Ribosome in the and ER endoplasmic reticulum ribosome and T. And then there could be some free ribosomes also. Cell now the cam ding in the day yung sivere kajishtinarwa. Echa ta the main beke ta wangbo plant cell now the keshu plant cells plant to get photosynthesis shigirwa. So chinken ki ta chigyan nime yo de so lenken chi gogurwa ta dile yang so plastid such sigirwa. So plastic tikin the plastic kajishtirwa. 통규 나고 장국이 튀겨 다디 세브 스타일 만들어. 지가 직자도 야샤브데 디게. So let's say this is a plastid and you can draw another plastid kashish 튀기 plastid 망부 용구르와. Look, this is another plastid. Now let us draw a mitochondria. Mitochondria stiginda. Mitochondria katiga mautiga. Yeah, mautigura mitochondria inda. Do stage in ra. Do stidanta yagujine. Do stage. This is the mitochondria. We do another mitochondria over here. That's another mitochondria. Chloroplast tisai, mitochondria tisai, nucleus tisai, endoplasmic reticulum tisai. I've pointed out few of the free ribosomes. And then what else can we draw? Tadi nanglo la ya detina karsigre Golgi body chigurwa Golgi body di khwatiga das det chigurwa Golgi body di duk stage ne nucleus ki thid wajlirwa Golgi body those in the Golgi body chigurwa. That's a Golgi body. Of course, Kesa Dundanguma de la Kinazu Tempa Sugu in the Zenegio Selgi Dupcha Sogo Sumjioba Tabunang Dupcha Sogo Sum de Carrere Shubina. Number one, you have got a cell membrane. Then what else do you have? Nanglo Chimbina, you have the cytoplasm. What else? The third one is your nucleus. This one cell membrane, cytoplasm, and the nucleus. These are the three main components of a cell. So there are cytoplasm. Cytoplasm. The cytoplasm da cytosol ni khares signi the w re chik rebe keba yorbe tissue chung. Cytoplasm in the the chazan of Sasun and labeling we have to label it properly. That's the most important condition. So then get taking the labeling chedi. labeling the sanga singe member che labeling khadu chi du chidan chi yugure rwa. As far as possible, labeling check up, so do not cross the line. Namgyun chigdwa, thick chigya, thick chigma, t cross chigdwa. So try to avoid this thing. Ta labeling chidunge chilo di inda. That's your cell wall. Plant cells to have a cell wall. And nanglo di devi ina, what else? Nanglo devi ina. We've got cell membrane. Yang na plasma membrane na nai tigdwa, cell membrane, plasma membrane. Ngi labeling chigye. Plasma membrane, cell membrane, then this thing is called the plasmodesma. Then this is your nucleus. The nucleus ya kransugi surtu nucleus logach tina digida kansa nucleus di nangla ya chashe mangbutwa nucleus ki chilo de la nuclear membrane che ani nuclear pore yo bacherwa tange de chazan te de beina ya saja shongsa marerwa nucleus surtu tina digire penange de chikta tisha urwa nucleus di chik chembo sugi I'm going to magnify it. Okay, let's say this is a nucleus. Nucleus and then nucleus then this is your nucleolus. Let's say this is your nucleolus, and ta dinang lo ya ngege deep igan shungutwa chala yanto madu chesa nucleus nang la ya nganzu chala chilon chala nang lo le ya nang lo chala chilo to ya de de yungurwa odi ta nuclear pore gi juni chigre. This is called the nuclear pore ikhung lagirwa. This is your nuclear membrane ani nucleolus che ani tinang ya you can have these chromosome. These are the chromosome. These are the chromosomes, and then you have the nucleoplasm. Genetic material chromosome, and these are the nucleoplasm. Nucleolus, nuclear membrane, nucleoplasm, chromosome, then nuclear pore. These are the chemosome. And endoplasmic reticulum. And then this is a large central vacuole, vacuole, vacuole. And then this is your mitochondria. This thing is your plastid. This thing is your Golgi body. Ani cytoplasm timidwa. Okay, tange didwachil tishana cytoplasm didwachil. Cytoplasm. Did it? So, this is a diagram of a plant cell, and these are the various parts of it. Label chishayo yanto en kran so practice shi tagi chi gande tisunwa. You can draw it. Tagena na kran so ra digre. Rumu yaku goe de chi kechum buchi nenga shire. So you practice it. Ra the yangyo kon chi practice shi kran so kar garcha she kar kartu. And the demong detail kan ya chance lokte na la zari keve lokte na mare. But still, if you're interested, you can label these parts. Yeah, there are parts label chishayo. Tadina nge chik mix e chiki dilab du chungs. Tanda di coaches di chikche plastid mitochondria Tingi chigya wangpo chungu plastic cell organelle. These are called the cell organelle. These are very special kind of a cell organelle. Mitochondria and plastid are special kind of a cell organelle. Why am I saying special? Karchinerisena krani kawane chungshabchiresena anzu prokaryote cell ne chungshabchire. Origin point of evolution di anzu plant cell na lo khandis level na thoma mebache chisu lebachilla worda. They are double layered membrane. They have got double layered membrane ni yore ro mixe gi ti ni larwa plastid da di ni larwa Quran so rang DNA yor ti nenga shibchire cell organ the keshigme mixe gi plastid da mitochondria ni la they have got their own DNA ti yam sambuchire so ti DNA Quran ni la so rang DNA di yo sanga ro chik tati khawane chungpare se di ta chechu chebi na prokaryote cell ne chungshabchire. A plant cell is a eukaryotic cell, right? Because it has got a well-defined nucleus. Nucleus pe mixe same shudwa nucleus tempo shudwa. So this is a eukaryotic cell. Eukaryotic cell nang la mitochondria ta plastid ni di thoma mebache an chisu ya prokaryote cell chikta bacteria do chik eukaryotic cell na lep ani ko phegeran la eukaryotic cell gi prokaryotic cell di chame matang bache 아니 베게랑 라 네버체 펜토에 레가디 모투네 드룹데 버체 페나 플라스틱 기 펜토에 레가 지기로와 직가 가르세레 니여제조 토니야 직 슈가 널추디게 디스토 투고로와 소 펜토구도와 펜토구에 나 아주 잘라데 냐디기로와 소 디 theory 지글라 그래 딜이야 symbiotic theory endosymbiotic 티베나 this theory is called endosymbiotic theory endosymbiotic theory like endosymbiotic ngazu symbiotic jaimba sire jewaduso tumbar jewachilla ngazu type of nutrition nanglo ya ane symbiotic nutrition ya yorba chikben chiro chebade symbiotic na la ngazu nangse yebachemba parasitic symbiotic da de chechne so this is endosymbiont krani ja endosymbiont re kar sugar se na thoma krani prokaryote bache an chisu eukaryote cell na la lebache an duksche chen ane 다대차리야 아니 즉 eukaryotic cell 낭라 즉 내가 계신 부시지 제대고 그래 so 데지 라그래 아주 더 믹스기 크라닝에 슈거에 좀세 they have got their own DNA and they have got double layered membrane ngoene khurala membrane chi yobache chisu eukaryotic cell na lebdu ani ya membrane sa chira bache double layered membrane duk sha bache ani theory dilaya endosymbiotic theory like konzu prokaryotic cell ne chongba de시냐고 theory dik 어두시 다 remote practice 시로치 도시 당에게 remote cancer 차수나 다 animal cell 지기 믹스기 plant cell 야 쏟다 열다 rectangular shape 도 직체 믹스기 it has got a definite shape animal cell 야 shape mixes ke yo mare irregular 도시래 송에데 다 디티사수나 자도 종다 제래 디르와 가조 망아 종다 제비나 디그시 리모 야가 두그래 봐 라발이야 라고 상 두그렌데 네 키란소기 리모 야구 차수나 케벤시부 요래 예다기 슈스와 웹나라야 키란소 if you're interested annotated drawings in a karire how can it help you in learning the concept 롭존 셰베캅소인데 리모 균에 장토에 망부치운 그래 라 리모티브 참 많이 봐 리모리아 아주 노트스도 sambal이야 and kiranso dindeke you can make use of it annotated drawing the learning style chigre you can make use of it so go and look out for it in the web can see that can check you'll get some idea that delia plant cell sabache I've drawn the plant cell now animal cell chitigin darwa do sheshinda 레로시 animal cell 들어와 딱 통계나 도시 되네 도시네 도시네 다디 루모디 키난조야 고고로시 야고 친수나 디기르와 난저기 도시네 디징에 3D dimensional 도시 계절이 다디 Tidanta damida yungure you can even draw a very simple diagram of animal cell but ine dirwa nga joge remoning chebo stubina soso ina ya lobsen ya changege doba diligi dwa ing shugu dwa or the cheduni. Dokshe and tenenge. Now I'm going to draw the nucleus. Nucleus the key to Sherwa. Like this. This push it and then dokshe like this. Then let's say this is your nucleolus. That's your nucleolus. Now, these are the nuclear pores found on the nucleus. And you can highlight some nucleoplasm. And of course, your dense chromatin material. Those shit. That in that shemba taking on the plant cell na draw shishaba wangbu chungu kachigyoba cell organelle that we have drawn in case of a plant cell. We're going to draw the same thing again. Now, for example, I can draw endoplasmic reticulum. Like this. So I can draw endoplasmic reticulum like this. I can even draw Golgi body. We have seen that thing in plant cell. Like this Golgi body, we can draw Golgi body. And then what else can you draw? There is another thing called lysosome. Let me draw this. Lysosome, let me draw a few of those. Lysosome. Lysosome plant na la mes sugar rwa. Ta ine tsani pa khachigi plant richik na lo lysosome yungres sugar, but not in all of the plant cells. So lysosome the mixing animal cell gi khechuchre. I'm going to label it near Jul chawra. Label the lysosome is there in animal cell, but generally not found in plant cell. What else can I draw? Of course, one of the most important thing is, and in animal cell there are centrioles. So centriole chawra. Centriole are found normally near the nucleus. Nucleus ki thi dwa yorirwa. Let's say this is the centriole. Let me draw another centriole. Centrioles plant cell nala yomare, centrioles singe the animal cell nala yore. Azu jula kebar tibi kapsul libgir dati gedi. Then what else? Of course, mitochondria ti chogre ngazu. Mitochondria ti chogre. Plant cell nala we have drawn a plastid, but in animal cell it's absent. Animal cell nala plastid yomar because animal cell they don't photosynthesize. So plastid mevache. Mitochondria kashish tine degree. You can draw a few of those mitochondria here also. Taking issues mitochondria ta plastid digeti endosymbiotic race, endosymbiotic noma prokaryotic cell ni chungsha race, and that they have got their own DNA. DNA rangi DNA yor. No other cell organelle have their own DNA except mitochondria and plastid. There are few vacuoles, small size vacuoles. Only vacuole the TV na. Let me draw vacuole. Vacuole chung kashish tiora. And then of course free ribosome then kashish tigo. Dikana yadang ribosome ya tjedwa. Endoplasmic reticulum kala. So endoplasmic reticulum kala ribosome lebkire that's called the rough endoplasmic reticulum. This shit ani really chunju dozo ribosome ani ribosome kha chigi ya free floating ribosome, free ribosome inda. free ribosome kachik che do che ta de ne nganzo ge tamatela cytoplasm tv na let's say these things are the cytoplasm. So how about this diagram of an animal cell? 독세 땅 안쪽에 레벨링 지체로 봐. Labeling chilo dilia cell membrane. We don't have cell wall in case of animal cell. Chilo dilia on the cell membrane yore chingi. Then let's say this is your mitochondria. Mitochondria. The one that has got its own DNA. DNA susurang DNA yore. And this is your endoplasmic reticulum. Endoplasmic reticulum smooth re rough re cheche. I'm just mentioning endoplasmic reticulum. Cherwa. And what else do I have? These are the vacuole. Animal cell nala vacuole yorte ine vacuole chungju ne de mangbo to cap de yurta. Chili manga de yong sibre. A vacuole size chungju re. But in case of a plant cell, the vacuole was bigger in size. It occupies near about 95% area of the plant cell, 90 to 95% of area of the plant cell. But animal cell, they are minute in size. They are small in size. Dukshe yore mangushi kyab de yungure in a size chungjure chejungde. Then what else do I have? Let's say these are the free ribosomes. Ribosome. Then this is the nucleus. Taking a nucleus ki kola yamas detail gi remote taking ingo the tisa bingi wa so nge de tim moko bache yangya. So I'm just labeling it as nucleus. Tokshe and then of course nge de ha this to centriole. Centriole. That's one of the very important organelle in case of animal cell. Tokshe centriole. Then cytoplasm. Tokshe what is Golgi body ladwa. Golgi body. Golgi apparatus la nahi tik tu kashi body tikre. Let me write apparatus. Golgi apparatus. Keba yamare body apparatus. Tokshe ta karla su shemba lysosome of course the lysosome la itlaswa. Lysosome. Animal cell it's a bit irregular in shape. Now we are going to differentiate between plant and animal cell. I will tell few differences between plant and animal cell. So let's discuss what are the basic differences between plant and animal cell. So first thing, plant cell is chair because of that cell wall and large central vacuole. Animal cells are a bit smaller in size. Size ki chejung ton tesi keba yung digre shemba keba kartu sevi na sopta gitone. Plant cell has got a particular shape, whereas animal cell doesn't have a particular shape. Mikse ki sopta skeyo mare it may change rwa the deche dichitangi keba che ni ki phala ya. What else samdam be na major differences nga zo typical so katu ina plant cell the animal cell ni pala keba the typical so katu ina sam thanshu de kartang gugre. What are the major differences? And one of the major differences is, in case of a plant cell, it has got a plastid. Plastid yorda. Plant cell nangla plastid yore. Animal cell nangla you won't see plastid. Plastid yor mare because animal cell do not photosynthesize. Plant cell does. So do chechinirwa. Plastid. Ta kiran so ya plastid la rik yungure. Plastid la ya. 당에 소브 릭숨 라파 쉐비나 지단 릭 시버지야 용구르데 이네 숨 라파 소브 숨 딜라야 chloroplast nam yu khode chre chloroplast ni patelaya khatika chloroplast chila chromoplast chro chromoplast Then dingibat sumba de la leucoplast. So these are the types of plastid. If you could know that it's good for you. Plastid ki la yawa sum yungure ta chloroplast sena karere lana ngazu tante de jane de chloroplast sena lana. It's a plastid that has a chlorophyll pigment. Rawa photosynthesize chinge di sure plastid rixum nan de la ya musum dugudwa ya chloroplast plastid ni di paliya. Plastid ina chloroplast im khabu marwa. Plastid ina chromoplast ya yisivre leucoplast ya yisivre. Chloroplast ina plastid im khab dugudwa. So phakep surkhab diya yan samdan wa. So chloroplast ti chobu khande chloroplast is a plastid. Chloroplast is a plastid. Plastid kadichrela na that which helps in photosynthesizing because it has got chlorophyll in it. So chloroplast na chlorophyll jangze di yosa it can fix sunlight and do photosynthesis. So the naturally ya chloroplast. Chromoplast again it has got pigment yorta but chloroplast na pigment kadu chlorophyll pigment dua. Chromoplast na pigment kariore subina the carotenoid pigment singing the euro. Medo color harpo in the midway. Shingdo git sendo mandamanda arm serpo so the kushu get the marble in the sable in the jungle within the metal sonda ning chebo dama digit sonda colorful petals are there. So this hangma chromoplast ki bekire chromoplast ki pendo kariore chromoplast ki color pigment carotenoid singing the use angarta. So carotenoid ki pigment the pendo kariore sebina tada medo color harpo yogo in the karre so that insect may come and lebina budik lebina drama digeti metokal khorgidwa tike gi pento khariyolana they help in pollination ro metodigi tepa chik khasirenga jogi pollen grain digeti pha khap thoya medo sheng stigma gala phapchinen dukshe fertilization chechane yungruba process di maso sexual reproduction di ta ti yongpala ya pollination ko eti kechushirwa chagen tangbodi chochishudi and pollination chinkenti bees digedire wa medogi damade cholo coaching colorful petal they attract the insects and insect levina budiged levina that will help them in pollination. So chromoplast ki pento de uchire mixege de chromoplast ki near the photosynthesis deva ke yo mare photosynthesis the deva yo badi chloroplast re chromoplast only helps in pollination wa de deche ngajoi ne meto damaning je bu to ba yi thogo de chagdwa so that gives us pleasure also. Leucoplast the mix the pigment major. The dingy na pigment doa. Chloroplast has got chlorophyll pigment. Chromoplast has got carotenoid pigment, but leucoplast is the only plastid that does not contain any pigment. Leucoplast na pigment kyun yomare. And the nala protein da the lipids to store shell pentogore. What did do chi plastid color? So mix plastid kwayore shubina plant cell na yore. Animal cell na yore belana yo mare chue din ashingi any kebarchik kardu plant cell na taking issues wa it has got a large central vacuole the chembudwa kilaya around about 90% area is covered by vacuole. It's a very large thing and the lega ya mangbu shibu chego vacuole gi ra check kasigre nga so plant the cell the rigid soya it acts as a storage also. Plants vacuole the storage here, water storage here, molecule breakdown here, la complex molecule the breakdown here, la pentogore, waste material storage here, la pentogore, and then there is another thing called lysosome. The plant cell na la yorebe lana lysosome timaso angi. Plant cell na lysosome yo mares chiche. Rawa chitang geshigyoda. Chitang generally plant cells do not have a lysosome. Goyase ke yo mare kumsenti lysosome ki function di kharsena ngajogi enzyme di gedri release che chikche ani phagocytosis regre check ngajo kharsigre it's a namgyune ngajo dile ya ming shemba chi pyogi suicidal back chechne phagocytosis chigyo be ke girwa cell chi cell shemba chi zame tamba engulf cheva dila phagocytosis legirwa ho den chik lysosome ki lenge de de uchre ngajo foreign body dige leve na konzo engulf chechne zame ta ya da dige di tachik phagocytosis ki legachi indu ani di chinke de lysosome rwa tadi animal cell na yorda but in case of a plant cell dide goya yo mare lysosome la ne surdu mixegi wangpuchi chunghuchi goya mendwa kansna lenge de changma sui dub gore sena vacuole rang de dub gore so ngone yogi chikla ya kirala ya pen shanyu yobe na shanyu chi remote ya di remote plant cell animal cell gi remote ya shanyu yobe na kirala ya shanyu yem tebachi gogudu sebe na ani gogu mendwa 양계치 가진 고구래 since you have got a pencil, you don't need another pencil. So dinang shirwa vacuole gi lega chasan chigyu na rwa tanda di storage de na enzyme release ya ta mangbu chig lega di phagocytosis gi lega di chasan chigyu na ani wangpo shimba ji goye mendwa. That's why plant cell do have lysosome, because the function of lysosome in plant cell is done by a vacuole. This way. So plant cell, the animal cell, plant cell have got large central vacuole, whereas in animal cell there is no such thing. Vacuole yorte, but they are small in size. Mangbo che chungjung, mangbo chungjung. Animal nala vacuole di mangbo yorche chungjung. In plant cell, one large central vacuole. The kebarchi. Then shamba kebar kardu, yes, here. Animal cell nala we've got centriole yorda. Plant cell nala central yoma. Central goese ke yomare. Central zitsobo karre che to race and central is mainly for cell division. Cell divide che kapsu pechu chimbu yordi. Plant cell nanglo la central goese kaisana microtubule senge organ chiyor. Kogi che shogur. So microtubule di organ di yogi la central goe mendwa. What did it uchre? So keba di che central kawa yore animal cell nal yore. Plant cell nal yeah, do you find centriole? No, absent. So the degi kebar chik che. Tene shemba kebar tere ruchre. So nge dele ya golji body ni chal yore rwa. Plastid di chik kebar chungutwa. Lysosome di kebar ni che. Centriole di che. Vacuole di kebar che. Ane ta kebar shite de mixe rimuna tonswa. Odi che. Ane chejungu ne kebar chito na digre. 대직체 독체 다 개발디 안 sobdegi tone 개발지 되나이 디그레 plants animal cell mixegi sobdaske yo mare definite shape yo mare plant cell it has got a definite shape rectangular shape doche particular shape jure 아니 체중기 tone plant cell 데시 체체 animal cell 데시 shuma doche 아니 드네 shema major differences dige di detne vacuole gi tone 개발 centriole gi tone 개발 plastic gi tone 개발 lysosome gi tone 개발 든테기 토네 개발체 오독시 아니 개발돼서 찾아지 마 제때 차비 르모야 티차비 켜서 르모디 프랙티스 야고 체로치 안 르모디 야고 셴베이나 안 켜서 개변 요래 롭센디 데스틱 잉슈야다 아니 간제 토네 남균에 대셔 개비값수 든테 paragraph 든데 링부들이 팀하고 버치 you can take the help of a well labeled diagram and you can even work on annotated drawing skill 디안 learning style chigre, so you work on it and it will help you. 독시 아니 게 타마들 개발된 비 장마 아니 믹스에 기치기 키란솔라 information ching에 대교디 가래 mitochondria and plastid they are special in a way unique culture special culture in a way that they have got their own DNA right and chronic origin evolution di cv na chunggung di kandes chungpare eukaryotic cell nala plastid da mitochondria singe di kandes chungpare seve na di deshe ge di theory chi deshe ge and that theory is called endosymbiotic theory and therefore chronic langazu mixe gi ming chitaga endosymbiont so these are called as mitochondria che plastid ni these are the endosymbionts 독세 제가비 다리델라 아니 배제 그래서 고수입을 해봐요 아니 디간라 독티들 여비나 아니 저와 그렇지 도시나